Hello students, greeting of the day. Myself Srimati Gitanjali Mahapatra, lecturer in zoology, both Panchayat College, both. Today I will discuss about the topic spermatogenesis. It is in plus 2 second year syllabus. Here we will discuss about gametogenesis, spermatogenesis, stages of spermatogenesis, formation of spermatids, spermiogenesis, stages of spermiogenesis, structure of sperm, hormonal control of spermatogenesis, then significance. Before going to discuss about spermatogenesis, we should know what is gametogenesis. Gametogenesis is a biological process of formation of haploid gametes. Haploid gametes are sperms and ova. These gametes are formed from the diploid primary germ cells. Those cells are called gametogonia. Those are named as spermatogonia in case of males and oogonia in case of females. Those are present in the primary sex organs called gonads. Gonads are of two types. The gonad in case of male is called testis, and in case of female the gonad is called ovary. Now the gametogenesis is of two types. First one is spermatogenesis, second one is oogenesis. Then come to spermatogenesis. Spermatogenesis is the process by which haploid spermatozoa or male gametes develop in the seminiferous tubules of testis. Testis contains a large number of small compartments. These compartments are known as testicular lobules. Each testicular lobule consists of 2 to 3 seminiferous tubules. Each seminiferous tubule is lined by a layer of germinal epithelium, the cells of which undergo mitosis to form two types of cells. One type is called stem cells, another type is called spermatogonial mother cells. Spermatogonial mother cells means the cells which produce spermatogonia. Those cells are known as spermatogonial mother cells. The stem cells are those cells which produce spermatogonial mother cells phase by phase to produce large number of sperms throughout the adult life. So, the stem cells remain near the basement membrane or basal membrane of seminiferous tubule to produce sperms throughout adulthood. And the spermatogonial mother cells undergo spermatogenesis to produce sperms. The mother cells undergo spermatogenesis to produce the sperm. In this diagram, you can see the different parts. This is the longitudinal section of testis. This is one compartment. This compartment is called testicular lobule. This compartment contains 2 to 3 tubule-like structures. These are called as the seminiferous tubules. This is the, now come to the cross sections of the seminiferous tubule. This is the transverse section of the seminiferous tubule. This is lined by the cells. These are known as germinal epithelial cells. The germinal epithelial cells undergo mitosis. They undergo mitosis to produce stem cells and spermatogonial mother cells. They are spermatogonial mother cells undergo spermatogenesis to produce sperms which are present in the middle. Now this is the enlarged view of a section of the seminiferous tubule. In this case, you can see this is the covering of the seminiferous tubule. This is the basement membrane. These are the diploid germinal cells which undergo mitosis to produce primary spermatocytes. Primary spermatocyte is diploid. The primary spermatocyte undergoes meiosis 1 to produce secondary spermatocyte. Secondary spermatocyte undergoes meiosis 2 to produce spermatids. The spermatids change to sperms or spermatozoa, which are present in the center of the seminiferous tubule because it is the enlarged view of a part of the seminiferous tubule. So, this is the process of spermatogenesis which you can see in this diagram. These are the different stages of spermatogenesis. Now come to the process of spermatogenesis. The spermatogenesis process involves two steps. First step is known as formation of spermatids. Second step is known as spermiogenesis. Formation of spermatids further involves three phases. First phase is called multiplication phase. Second phase is growth phase. Third phase is called maturation phase. First, we will discuss about the formation of spermatids. It involves three phases. Number 1, multiplication phase. Number 2, growth phase. Number 3, maturation phase. First come to the multiplication phase. The multiplication stage is the first stage in the formation of spermatids. Here, the spermatogonial mother cells present in the seminiferous tubule undergo mitosis and form a large number of spermatogonia. The mother cells undergo mitosis. Mitosis means the chromosome numbers remains same. So, mother cells are diploid, the spermatogonia are also diploid. Then, growth phase. The spermatogonia increase in size by accumulation of nuclear material and attain maturity. Then those are called as primary spermatocytes. After growth, the spermatogonia are called primary spermatocytes. In this structure, you can see this is the spermatogonia. Gonium singular, it is called spermatogonium. It undergoes mitosis to produce primary spermatocyte. This is diploid. Now the primary spermatocyte is formed, which is after the growth of the spermatogonium. Spermatogonium undergoes growth to form the primary spermatocyte. The primary spermatocyte undergoes maturation division to produce the sperms finally by the process of meiosis 1 and then meiosis 2. Then come to the maturation phase. During maturation phase, the two maturation divisions occur. First division is called meiosis 1, second division is called meiosis 2. Meiosis 1 is reduction division where the chromosome number is reduced to half. But meiosis 2 is equal division where the chromosome number does not reduce. During maturation phase, the two maturation divisions occur. First, each primary spermatocyte. The primary spermatocyte is formed after growth phase. The primary spermatocyte divides to form two secondary spermatocytes. This is the reduction division. This is called meiosis 1. So, the two secondary spermatocytes which are formed after reduction division are haploid in nature. The primary spermatocytes are diploid, but the secondary spermatocytes are haploid because these are produced by the process of reduction division, which is meiosis 1. Then another, what happens to the secondary spermatocyte? The secondary spermatocyte, it divides by equal division where chromosome number does not reduce. That is called meiosis 2 to form two spermatids. Each secondary spermatocyte divides to form two spermatids. These secondary spermatocytes are haploid. The spermatids are also haploid. In this way, 4 spermatids are produced from each primary spermatocyte. In this figure, in this animation, you can see the formation of spermatids and spermatozoa. This is the from the outer surface, new cells are formed. This is the outer surface. The new cells undergo mitosis to produce the primary spermatocytes. Primary spermatocytes are converted to secondary spermatocytes. Secondary spermatocytes are converted to spermatids. You can see this in this animation. Let's come to spermiogenesis. By the process of spermatogenesis, each primary spermatocyte produces 4 spermatids. Primary spermatocyte is produced from spermatogonium after growth phase. Now the primary spermatocyte divides by two divisions. First one is meiosis 1, second one is meiosis 2 to produce 4 spermatids. What happens to the spermatids? The process by which rounded non-motile spermatids, the spermatids are round and they are non-motile, they cannot move. So, the rounded non-motile spermatids are converted to active mobile or motile sperms by the process that is called spermiogenesis or spermioteliosis. So, by the process of spermiogenesis, the non-motile rounded spermatids are converted to mobile or elongated mobile sperms. You can see the structure of sperm in this diagram. In this diagram, this is the sperm. This is the motile or it can move from one place to another place with the help of its tail. What happens then to the spermatid? The nucleus of the spermatid shrinks and chromosome become condensed, closely packed. The chromosomes become closely packed. The nucleus shrinks. All nuclear material get lost except DNA. What is lost? Generally RNA, proteins. These materials are lost during spermiogenesis, but the DNA is not lost. DNA remains in the condensed form. Now the sperm nucleus becomes elongated and narrow. The two centrioles move to lie behind the nucleus. All mitochondria spirally arranged in the middle piece of spermatozoa. The Golgi apparatus, which is present in spermatid, that forms the acrosome in front of the nucleus. The acrosome is present in front of the nucleus, which contains the enzymes. Those enzymes are called as the sperm lysins, which are responsible for the movement of the sperm through the egg membranes. The sperm lysins or enzymes are helpful for the formation of a pathway in the membranes of the egg during fertilization. Now, most of the cytoplasm of the spermatid are eliminated, but the remaining little amount of cytoplasm forms a condensed layer surrounding the middle piece. Very little amount of cytoplasm is present in sperm, but the spermatid contains large amount of cytoplasm. So, most of the cytoplasm are eliminated during spermiogenesis. Very little amount of cytoplasm is remain that is present as a condensed layer surrounding the middle piece. This is all about spermiogenesis. After the completion of spermiogenesis, the spermatids are converted into sperms. This is in this diagram you can see different stages of spermiogenesis. In the first diagram, this is the spermatid. It becomes changed to in the following stages: number 1, number 2, then to the number 3, then come to the number 4, then number 5, number 6, then number 7. In the following stages, the spermatid is converted. This is the spermatid. This is converted to sperm. This is the sperm, which contains 3 parts: head, neck, then this is the middle piece, this is the neck region, then this is the tail region. These are the different parts of the sperm or spermatozoa. Now, this is in this diagram you can see the total structure of the total figure about spermatogenesis. In this diagram you can see the different stages. This is the germinal epithelial cells. These are the stem cells. The stem cells undergo mitosis to produce spermatogonial mother cell. The mother cells undergoes growth to form the primary spermatocyte. Primary spermatocyte undergoes meiosis 1 to form 2 secondary spermatocytes. Each secondary spermatocyte undergoes meiosis 2 to produce 2 spermatids. In this way, 4 spermatids are formed from one primary spermatocyte. All the cells are attached with each other with the help of cytoplasmic strands. These cells are not separated from each other. They are connected with each other with the help of cytoplasmic strands. Now, the spermatids, they undergo spermiogenesis. By the process of spermiogenesis, the rounded spermatids are converted into elongated sperms. In the first case, the sperms are also connected with each other with the help of cytoplasmic strands. Now the sperms become free and they become free from each other to form the functional spermatozoa or sperms. It is the diagrammatic representation of the spermatogenesis. You can see also the different stages here. It is the first stage, it is the stem cell. This one is stem cell. Next, number 2, this is the spermatogonial mother cell. Then come to this is the primary spermatocyte. This is the secondary spermatocyte. These are spermatids. Then these are the sperms which are getting attached with the wall of the Sertoli cell. Now these are the free sperms. This is the diagrammatic view of the spermatogenesis. Now come to the sperm. By the process of spermatogenesis, the sperms are produced. Now come to the structure of sperm. What are present in sperm? The sperm, you can see in this animation the sperm, how the sperm moves. The sperm, it has two parts. One is head, another one is tail. This is the head of sperm, this is head, and this is the tail of the sperm. The head, it is triangular in shape and contains a nucleus and an acrosome. The acrosome is a double-layered cape-like structure. The acrosome is a double-layered cape-like structure containing enzymes called sperm lysins. The nucleus contains concentrated DNA. Two things are present in the head. One is acrosome, another one is nucleus. Acrosome is present in the front of head. Just behind the acrosome, the nucleus is present. The acrosome contains small sperm lysins, these are the enzymes, and the nucleus contains the concentrated DNA. Now come to the tail. The tail is the longest part of the sperm which provides locomotion to the sperm. It is divided into 3 subdivisions such as neck, middle piece and end piece. What is present in the neck? The neck is the joint portion between the head and middle piece. The neck contains 2 centrioles. One is proximal centriole and another one is distal centriole. The proximal centriole remains in the neck region, but the distal centriole extends through the middle piece and tail as a filament. That filament is called axial filament. The axial filament contains 9 double peripheral fibers and 2 single central fibers. This is called 9+2 arrangement. Next come to the middle piece. It is the second part of the tail. The middle piece bears axial filament surrounded by tightly coiled mitochondria. The mitochondria supply energy for the motility of the sperm. The sperm, in this figure, you can see the sperm moves. This movement requires energy. The energy comes from the mitochondria which are present in the middle piece of the tail region. Now come to the end piece. End piece is the last part of the sperm tail. The end piece contains axial filament surrounded by a sheath that is called tail sheath. Now this is the figure of the sperm. In this figure you can see this is the head, this is the acrosome which contains sperm lysins, this is the nucleus which contains concentrated DNA, this is the neck region, this is the total tail. The first part of the tail is called neck, second part is middle piece, last part is end piece. The neck contains the centriole, this is the proximal centriole, and here the elongated axial distal centriole is present which forms the axial filament. Around the axial filament in the middle piece, the mitochondria are spirally present. This is the mitochondria. Then now come to the tail. The elongated tail end piece. It contains axial filament surrounded by a membrane that is called tail sheath. This tail moves by which the sperm also moves to reach the egg. This energy is provided by this mitochondria. Now come to the hormonal control in spermatogenesis. How the hormones control the spermatogenesis process. Mainly hormones are responsible for spermatogenesis. So at a particular age, after attaining a particular age, this spermatogenesis occurs. In children, this spermatogenesis does not occur. So the hormones are secreted at a particular age, after which spermatogenesis starts. The main hormone is known as gonadotropin releasing hormone, which is secreted from the hypothalamus of brain. Brain contains hypothalamus, which secretes the hormone called gonadotropin releasing hormone, which is simply called GnRH. This GnRH, which is secreted from hypothalamus, stimulates the anterior pituitary gland to secrete LH and FSH. LH is luteinizing hormone, FSH is follicle stimulating hormone. These two hormones are secreted by the anterior pituitary gland after stimulation by GnRH of hypothalamus. At a particular age, hypothalamus secretes GnRH. This hormone stimulates anterior pituitary gland. Then anterior pituitary gland secretes LH and FSH. What is the function of LH and FSH? LH, first come to the LH, that is the luteinizing hormone. It acts on the Leydig cells of the testis. Leydig cells are the cells which are present in the interstitial space. Interstitial space, which is the space among the seminiferous tubules. Among the seminiferous tubule, the testis contains small spaces. Those spaces contain the cells. Those are called interstitial cells or Leydig cells. These cells secrete testosterone by the influence of LH. Now this testosterone, the role of testosterone is to stimulate spermatogenesis. Testosterone stimulates spermatogenesis in the seminiferous tubule. Now what is the function of FSH? FSH is another hormone that is secreted from the anterior pituitary gland, but it acts on the Sertoli cells of the seminiferous tubule. Sertoli cells are the special cells. Those give nutrition to the developing sperms. These Sertoli cells are elongated cells. They are present in the seminiferous tubules. Those cells are stimulated by FSH. FSH is produced from the anterior pituitary gland. Then the FSH stimulates the Sertoli cells of the seminiferous tubule to secrete an androgen binding protein and inhibin. Two things are secreted from Sertoli cells. First one is androgen binding protein. This protein concentrates testosterone in seminiferous tubules. Seminiferous tubules concentrates testosterone to stimulate spermatogenesis, which is by the influence of ABP. Another hormone that is known as inhibin, it is also secreted from the Sertoli cell. It suppresses excess FSH synthesis by negative feedback mechanism. What is negative feedback mechanism? When the concentration of FSH increases, at that time the inhibin stimulates the hypothalamus to secrete less amount of GnRH. Then this also stimulates the anterior pituitary to secrete less amount of LH and FSH. In this way, the testosterone secretion decreases. FSH secretion decreases. This is known as negative feedback. FSH acts on spermatogonia. What is the role of FSH? FSH also acts on spermatogonia to stimulate sperm production. So FSH stimulates the Sertoli cell to produce ABP and inhibin. Those do their functions. FSH also stimulates the spermatogonia to produce sperms. Now this is the diagrammatic view. Here you can see this is the first case you can see this is the hypothalamus which secretes GnRH. It stimulates the anterior pituitary gland. Then anterior pituitary gland produces gonadotropins. Gonadotropins means LH and FSH. These two are known as the gonadotropins because they stimulate the gonads. They act on the gonads. Gonad is testis in case of male. So these two are known as gonadotropin. Now LH stimulates Leydig cells. LH stimulates Leydig cells to produce androgens, and FHA stimulates Sertoli cells to secrete the factors for spermiogenesis. So, this is the diagrammatic view of the hormonal control of spermatogenesis. Now, come to this is also the diagrammatic feature of the hormonal control. In this case, you can see this is the hypothalamus secreting GnRH. This is the pituitary gland. It is the anterior lobe. This one is the posterior lobe. This anterior lobe, GnRH stimulates the anterior lobe. The anterior lobe secretes LH and FSH. The LH and FSH, they stimulate the LH and FSH, they stimulate the seminiferous tubule. FHS stimulates the Sertoli cell to secrete the ABP, androgen binding protein. Now this ABP concentrates testosterone, then LH also secretes testosterone. This is the diagrammatic view of the spermatogenesis. In this case, you can see the negative feedback mechanism. The inhibin hormone stimulates the hypothalamus to secrete less amount of GnRH. Finally, FSH concentration decreases because pituitary gland secretes less amount of FSH. When this happens, when the FSH concentration increases in the testis or seminiferous tubule, at that time it automatically regulates by the hormone inhibin. The secretion of FSH decreases automatically by this hormone. This is called negative feedback mechanism. Now come to the significance of spermatogenesis. What happens? What is the role of spermatogenesis? During spermatogenesis, one spermatogonium that is diploid produces 4 sperms. The sperms are haploid. The sperms have half number of chromosomes. After fertilization of the ovum, the zygote is formed. The zygote is diploid, so it maintains the chromosome number of the species. The cells of the species remain diploid, but the gametes are generally haploid. So after fertilization, they can form the diploid zygote. The zygote, by continuous mitosis, can develop into an organism. The zygote is diploid, the organism is also diploid. During meiosis, crossing over takes place. During crossing over, there is the exchange of genetic material, so it brings about variation. During crossing over, the genetic material between 2 homologous chromosomes occur. Exchange of genetic material between 2 non-sister chromatids of a pair of homologous chromosomes occurs, due to which the variation appears in the next generation. It results in evolution. Next, spermatogenesis occurs in various organisms, so it supports the evidence of the basic relationships among different organisms. This is all about spermatogenesis. Thank you all. Have a good day.